Hello and welcome. This video is about analyzing substances, and we are looking at a particular method called chromatography, and this is for C2, Unit C2 for GCSE Science. So the method is paper chromatography, and it's a method that can be used for chemical analysis, and it's used to identify, for example, additives in foods. And by additives, it could be something like artificial colors. So artificial colors, they can be detected and identified by this method called paper chromatography. And the way it works is as follows. You have a strip of paper, and on that strip of paper you put a pencil line, and there's your pencil line, and on that pencil line you put a spot of your sample that you want to analyze. And the next stage is then to put the whole thing into a beaker that has some solvent in it. So you take the whole strip of paper and you very carefully put the end of it in the beaker with the solvent, making sure that you don't put the sample into the solvent. If you did, for example, the whole solvent would be washed away into the solvent, and you don't want that. Let's just move this over a bit. So very carefully put the filter paper in your solvent. You put on a lid. Let's just label that. In fact, we've got our lid here. This is a solvent, and a solvent is basically a substance that can dissolve this sample here. You can't always just use water, because water might not dissolve it, so we sometimes have to use special solvents. The purpose of the lid here is to prevent evaporation. There you go, just tidied that up a little bit there for you. So the setup is as follows, and then what you do is you allow the solvent to rise up the paper. It will soak up the paper, and as it soaks up the paper, it will dissolve the sample and it will move the sample up with it. And what you'll find after a while is that the colors will separate out. So some of the colors will move quite high up, some will move not as far, and you end up with a pattern that looks a little something like this. Now the reason why these separate out is because some of them dissolve a lot in the solvent, or dissolve very easily in the solvent and move up very quickly. Some of them don't dissolve as well and move up slowly, and because some are moving slowly and some are moving more quickly, they separate out like so. Now what's actually the point of this? Once we've done that, how can we actually use that? Well, what we can do is we can compare samples. So here I've got another one. Let's just move that over a bit. And if we carried out the same procedure in exactly the same way, same solvent, same sort of temperatures and so on, and the same procedure, we could end up with a pattern that we can use to compare to. Now we can see for our second example, our second experiment here, the chemical that we have tested has two chemicals in common with our first one. It has this purple one at the bottom, and it has the green one nearer the top. So we can say that this sample here has two chemicals that our first sample had. Now that's all well and good, but we do need a way to identify and perhaps name what these substances might be. So let's have a look at an application of this process. So here I've got a piece of paper for our paper chromatography. I've got 4 samples, and we can actually label these as known samples. So we'll call them A, B, and C. And what we could say is there were additives in food. They could be colors, or they could be another type of additive, but these are ones that we actually know what they are. They come from a container that you've just labeled, and we know what they are. This one over here is our sample that we're not sure about. Let's call it sample X, and we want to identify and see what substances are in this sample X. So we're trying to see if any of A, B, and C is actually in sample X. So what we do is we carry out our chromatography as we've done on the previous slide, and after a while you end up with a pattern that might look a little something like this. Okay, so we've let it run for a little while, and the pattern ends up like this. Now what we can see here is that the sample does actually contain some of the known substances, and if you look carefully you might be able to identify there. Just have a quick look and see if you can spot which ones are in sample X. What you should notice is that this sample has some of sample A, or chemical A, and it has some chemical C. It doesn't have any chemical B. In actual fact, this is something that we don't know what that is. So our sample contains some A, some C, another chemical that we don't know, and it doesn't contain any B. So this is how we use chromatography to carry out a chemical analysis of substances that we might not know. We have to be able to compare our chromatography pattern to known patterns. So what you should be able to do is just give an outline of how this chromatography is done, how it works, and the most important thing probably is to be able to analyze results on what we call a chromatogram, which is something that looks like this.